The beauty of our region is unmistakable, though sometimes the forests and rivers can conceal serious problems, like an unwelcome companion, PCBs, a poisonous man-made substance in our soil, water, and air.
And the issue with PCBs is that if they get into the water of Spokane, they can bioaccumulate or collect in the fish, and if the fish are then eaten, it can cause health risks.
PCB contaminated soil can't be used for anything, and so oftentimes it's shipped down to a toxic waste facility where it's burned up, and at which point the PCBs are then just thrown up into the air to rain down everywhere else. So again, the problem isn't necessarily taken care of, it's just moved somewhere else.
So I've been trying to figure out ways to bioremediate or degrade PCBs in soil.
At North Central High School in Spokane, biology teacher Dan Shea has been working with students in a multi-year project that uses nature to reduce and perhaps even remove PCBs from the soil.
My role in the project is really trying to get the kids going and get them connected with the people that they need to, finding supplies and being a mentor in terms of the types of procedures that they want to do.
The project began in 2017 and has since been passed along to students like Hannah Fott, who along with North Central graduate Maylin Scott won first prize for their PCB presentation at the Washington State Science and Technology Fair.
My soil is collected out of storm drains out of Spokane, and then we bring the soil into the lab, and then I can run my tests on it. I have soil in containers, and I add fertilizer to them at the start, and then I aerate them or mix the soil every day, which helps the bacteria thrive. Then I just leave it alone, and the bacteria and fungi that is naturally in the soil will degrade the PCBs out of the soil.
We want our students to know that they can make a difference in the problems that are plaguing their place.
We discovered that we were able to take very high levels of PCB contaminated soil and degrade the PCBs down to non-detectable levels just using the native fungi and bacteria within the soil.
Hannah's project and this PCB project is one research strand among many that our students conduct.
In Hannah's project, she checks PCB levels by extracting DNA from soil in the lab.
So it's just a series of steps that I run the soil through a filter, and then eventually I just keep washing the soil until only the DNA, so I can figure out what species of bacteria and fungi it is.
It's not one organism that can do it all, that can break it all down. It's a assembly line. It's a factory in there that, one organism breaks down this part of it and one organism breaks down this part of it and this part, until then it becomes carbon dioxide that just flows out into the air.
I've always been very interested in microbiology and the small stuff in science. Through this project, I actually decided that I want to major in microbiology and go into research as a career after high school. This individual project that I've been running and that has been run by many other students throughout the year has shown me that I am capable of doing high-level research, and it just has given me confidence to go into that as a career choice.
It's a massive job field, especially with the focus on COVID and the testing and all of the research that's going into that. Biotechnology, even before this, was one of the fastest growing fields out there. And so we're really hoping that we're training that next generation of students to take on those jobs and solve those problems.
I knew I loved science, but this program has really taught me to be individual and confident in my own research, where I can take my science questions and how I can answer them just through research and through data and analyzing the data.
Projects like this are possible because of collaborations. Dan Shea is continuing the work of his mentor, former NC teacher Randy James, whose early efforts helped create North Central's Institute of Science and Technology. A 9,000 square foot classroom and lab facility opened in 2014. Over the years, science students have been working together and upon graduation, hand off their projects to the next generation. The PCB project is prospering because of cooperation among the classes, as well as partnerships and consulting with outside individuals and organizations.
My favorite part is just seeing them grow from young freshmen into, really confident college-bound seniors.
I come in every day, I just get to work. It's individual, I do my lab work, I do my data analysis, I write my papers, and it's all on my timeline, so it's really teaching me how to lead myself and how to complete research on my own. And it's just really encouraging to see promising results out of this project, and it shows me that I am prepared for research at the next level. After graduation, I'm going to go to Washington State University and I'm going to major in microbiology and hopefully complete a lot of undergrad and graduate level research there.
The research community in Spokane takes our students seriously.